Welcome to the Basics lecture on DNA structure. We have seen earlier that the structure of DNA is a polymer of nucleic acids. Because the structure of DNA is critical to how it functions, let's go into greater detail now. The backbone, shown here in blue, consists of the sugars and phosphates. The backbone always has a 5' end and a 3' end. 3' end is the sugar end, and the 5' end is the phosphate end. The nitrogenous bases are located in the center of the DNA strand. Notice there are 4 different types. Thymine and cytosine are considered pyrimidines, and adenine and guanine are the single ring purines. 2 strands of DNA can bind together with hydrogen bonds between the bases, but these only form if the bases match each other in a specific pattern. As you can see here, the purine adenine binds to the pyrimidine thymine, and the purine guanine binds to the pyrimidine cytosine. A pyrimidine always binds to a purine because 2 double-stranded rings or 2 single-stranded rings would be the wrong width for the double-strand DNA. Notice also that the 5' of one strand is always bound to the 3' end of the other strand. This is called the anti-parallel nature of DNA. In the cell cycle lectures, we talked about DNA replication, or how DNA makes a copy of itself. Let's look at that at the molecular level now. DNA is copied by separating its 2 strands and making a new strand to match each parental strand. We're going to skim over the process a little bit, but you can see that a protein called helicase is responsible for unzipping the 2 strands of parental DNA, and a protein called DNA polymerase creates the new DNA strand by adding nucleotides, one at a time, to the 3' end of the new strand. New DNA is always built from the 5' to the 3' end. In cartoon form, we can show this as the parent strands separating, and then new nucleotides being added to each parent strand so that each new DNA molecule is half old material and half new. Notice again that G's and C's are always found together, and A's and T's are always found together. The addition of new nucleotides to the growing strand always happens at the 3' end. The hydrolysis reaction breaks 2 phosphates off from the new nucleotide, as regulated by DNA polymerase. Then a dehydration reaction directly follows, binding the new nucleic acid onto the sugar of the 3' end of the nucleotide above. Now think about our lecture on energetics. Which of these 2 reactions, this one or this one, is endergonic, and which one is exergonic? DNA duplicates during the S phase of the cell cycle. You know that the duplicated chromosome contains 2 sister chromatids, shown here. You've now learned that within each chromatid, after duplication has occurred, one of the strands is original and one has been freshly added. The structure of DNA can change because of things like environmental damage, replication errors, or errors during crossing over. Some common types of mistakes in DNA structure are as follows. A deletion removes a segment of DNA. Duplication causes a repeat in a section. An inversion occurs when a section is removed and then replaced in the opposite direction. And translocation is moving a segment from one chromosome to a completely different chromosome. The most common mutation, however, is just a change in a single base pair on the DNA strand, usually occurring during DNA replication. This is called a point mutation. Lastly, in traditional biology, we talk about using DNA to code for proteins, and we use figures like this one that assumes that all DNA turns out to make all kinds of proteins. But what else is in DNA? If you consider this box to be all of the DNA in a normal human cell, genome studies have shown that only 1 or 2% of it is actually genes, or DNA that codes for mRNA that is used to transcribe proteins, which is much less than researchers thought. The vast majority of DNA seems to be regulatory sequences or that control genes, or sequences that transcribe RNA that is used for something else besides simply making a protein. So in our next lectures, we're going to talk about the traditional gene and how it makes a protein, but then we're going to expand that out to look at how complex the genome actually is and what some other mechanisms of regulating seem to be important in the DNA molecule. So let's define some important aspects of a DNA strand when we talk about genes. We've mentioned that DNA is anti-parallel, with the 5' of one strand being bound to the 3' of the other strand. The 3' to 5' strand is always the one that we consider the template strand when we're talking about transcription, which is this one right here. And the other strand is called the non-template strand or coding strand. The promoter region is where transcription begins, and the start point is where the first mRNA nucleotide will be attached. The transcription unit is the sequence of DNA that will be transcribed into mRNA. This completes our basics video for today. Hope that you can write out the answers to these questions and do well on the basics quiz. Thanks for watching.